Obtain your tube that was shaken vigorously for 5 minutes. Preheat the test tube heater to 55 degrees Celsius. Place the test tube heater block in the test tube heater. Place the shaken tube into the preheated test tube heater. Set the timer for 5 minutes. Obtain the tube from the test tube heater and put it in the 4-section test tube rack. Set up the centrifuge centerpiece. Take the tube and place in the centrifuge. Take the 5 ml test tube with red cap number 1 and place directly across from the other tube. Close the centrifuge and set the RPM to 10,000 for 2 minutes. Then press start. Remove the tubes from the centrifuge and place it into the 4-section test tube rack and turn off the quentrifuge and unplug it. Obtain your 100 to 1000 microliter pipette, pipette tips, magnetic cap cover, magnetic binding beads, timer, and tubes. Set the pipette to 200 microliters. Unscrew the test tube with red cap number 1 and place back on the yellow part of the 4-section test tube rack. Unscrew the shaken tube. Obtain a pipette tip. Pipette 200 microliters of the liquid and disperse in the test tube with red cap number 1. Set the pipette to 250 microliters and unscrew the magnetic binding beads. Obtain a new pipette tip. Take 250 microliters of the magnetic binding beads and place in the 5 ml test tube with red cap number 1. Let it incubate at room temperature for 10 minutes. Pick up the test tube with red cap number 1 and place the magnetic cap cover on top of the red cap. Shake vigorously and then invert on a flat surface for 30 seconds. Unscrew the magnetic cap cover and pour into the beaker. Set the pipette to 500 microliters. Obtain a new pipette tip and pipette 500 milliliters of wash buffer 1 to the now empty 5 milliliters tube. Put the red cap and magnetic cap cover on the tube, and then take off the cover only. Shake vigorously for 30 seconds to wash the magnetic beads. Put the magnetic cap cover back on the red cap and invert it. Set the pipette to 900 microliters. Take the magnetic cap cover off and pour it into the beaker. Unscrew the wash buffer to solution. Obtain a new pipette tip. Pipette 900 microliters of wash buffer 2 to the same empty 5 milliliters tube. Put the red cap and magnetic cap cover on the tube, and then take off the cover only. Shake vigorously for 30 seconds. Put the magnetic cap cover back on the red cap and invert it. Set the pipette to 100 microliters. Unscrew the magnetic cap cover and pour into the beaker. Using the 100 microliter pipette, remove as much of the wash buffer from the cap without aspirating the magnetic beads. Let the beads dry on the lab bench until they turn a light brown color for 5 minutes minimum. Once dried, add 200 microliters of elution buffer directly onto the dried beads in the cap. Transfer the cap with the rehydrated beads onto a clean 5 ml tube and remove the magnetic cap. Clean up and obtain the test tube heater. Incubate the elution tube with the suspended beads in the test tube heater for 10 minutes at 55 degrees Celsius. Once the 10 minutes are up, remove the tube from the test tube heater and place it on the 4-section test tube rack, and then turn off the test tube heater. Invert the tube and using a robust downward shake motion, transfer the eluted DNA to the inside of the cap. Put the magnetic cap cover on. Take off the magnetic cap cover and place on the 4-section test tube rack. Obtain the ASI storage cooler containing positive controls according to your desired pathogen. Obtain the ASI storage cooler containing master mixes according to your desired pathogen. Compile your supplies. Set the pipette to 100 microliters. Put the positive control tube into the yellow section of the rack. Label the ASI mix tubes with P, S, or N in place in the green section of the rack. Unscrew the positive control tube. Pipette 100 microliters of nuclease-free water to the positive control tube. Stick the pipette at the bottom of the tube and slowly compress and decompress the pipette until the bead has completely dissolved in the water. Allow the positive control tube to rehydrate on the bench for 5 minutes. Using the 22 200 microliter pipette, transfer 20 microliters of nuclease-free water into the last ASI master mix tube, labeled N. Recap the tube when finished. Using the same pipette with a new pipette tip, transfer 20 microliters of each sample into the appropriate ASI master mix tubes. Be sure to recap each tube immediately after the transfer and use a fresh pipette tip for each. Transfer 20 microliters of the rehydrated positive control into the first ASI tube labeled P. Vigorously mix each ASI master mix tube by flicking the bottoms of the tubes. Then use a quick downward motion to send the liquid to the bottom of the tubes. After 5 minutes of incubating, samples are ready to run through the AWTT device.